Pollination and double fertilization in flowering plants eventually lead to the formation and development of the seed. In this module, we will focus on the basic structures that are found in mature seeds and their functions. Seeds are the fertilized mature ovules that result from the sexual reproduction of plants. The embryo, endosperm, and seed coat make up the basic structures of a seed. In the succeeding slides, we will look at variable features of these structures using monocot and dicot model species. Let us start with the embryo. The embryo is the earliest form of the plant. The illustrations in this panel show us embryonic structure in a dicot and a monocot seed. In this dicot seed, we have here our embryo, and in this monocot seed, we have here our embryo. Seed embryos are composed of an embryonic shoot, root, leaves, and stems. Some of these structures may be called differently depending on whether the seeds are monocotyledonous or dicotyledonous. This table shows variations in the embryonic structures found in dicots and monocots. We have here the gross morphology of a germinating dicot seed, and right here is the gross morphology of a germinating monocot seed. The major difference in the overall morphology, as you can tell, is the presence of the cotyledon in the dicot, which is essentially the embryonic leaves. In monocots, the embryonic leaf is referred to as the scutellum. In both dicots and monocot seeds, embryos have an embryonic stem referred to as the hypocotyl. They also have the embryonic shoot, known as the plumule, and the embryonic root, known as the radicle. The main difference between a dicot and a monocot embryo is the presence of additional structures in monocots, like the calyptile and the coleorhiza. It should be noted, however, that not all monocots are going to have all these additional parts. In general, monocots have more variations in embryonic structure compared to dicots. As I mentioned earlier, the presence of additional embryonic structures may vary across monocot species. Just look at the illustrations of the embryonic cross-sections of two monocot plants that we have here. We have onion in the left and corn in the right. The embryos in the seeds of both species are encircled in red. Now the onion embryo is almost similar to the dicot embryo in having an embryonic shoot, stem, and root, but it differs to a dicot embryo in having only a single cotyledon, and that is the structure that you see here. To the right, we have an embryo composed of a plumule encased in a coleoptile and a root encased in coleorhiza. Additionally, the single cotyledon has been modified to form the scutellum, which is the structure that you see here. The scutellum is an absorptive organ that lies adjacent to the endosperm. Another major part of a seed is the endosperm. The endosperm is a specialized tissue that is unique to angiosperms or flowering plants. The endosperm stores food that fuels the growth of seedlings after germination. In some species like beans and peas, the endosperm is completely absorbed in the cotyledons at maturity. Recall the morphogenic events during embryogenesis in the dicot species Arabidopsis. A key structure that is present in a developing embryo, particularly during the torpedo stage, is the suspensor. The suspensor connects the growing embryo to the developing endosperm. With this connection, the embryo has a source of nutrition it needs to properly develop and mature. At the torpedo stage, the programmed cell death of the suspensor is initiated. Based on previous lessons, the degeneration of the suspensor during the torpedo stage occurs because at this point, the cotyledons have taken up most of the nutrition from the endosperm. Then at the mature embryo stage, the suspensor completely degenerates as the endosperm is completely absorbed into the cotyledon. Depending on whether or not seeds have an endosperm that functions as food storage, they can be classified as endospermous or non-endospermous. Endospermous seeds have endosperms that function as the main source of food. A good example of endospermous seeds are your cereals. The non-endospermous seeds, on the other hand, are seeds without endosperms. Examples of non-endospermous seeds are those of legumes and orchids. In the case of many legumes lacking an endosperm, the food is stored in the fleshy cotyledons of the embryo. The dust-like seeds of orchids are exceptions, as they totally lack stored food reserves. In the case of orchids, germination requires the participation of symbiotic endomycorrhizal fungi, which provides the fuel to the seeds to germinate. The size of the endosperms of many seeds is often not uniform and may be differentiated into areas of varying thickness. The endosperm in the Arabidopsis seed, for example, has only a single layer of cells that have relatively thick cell walls. This is a picture of a cross-section of an Arabidopsis seed. From this picture, the two cotyledons are easily visible, as well as the root, which is the darkly staining structure in the seed. The brown outline constitutes the seed coat. Now, if we zoom in on a part of the seed coat, we will see that sandwiched between the seed coat and cotyledon is the endosperm, which is composed only of a single layer of cells. So here is our seed coat or testa, and here is our cotyledon. In the middle of these two structures is the single layer of cells that make up the endosperm. In contrast to Arabidopsis, beans have larger endosperms. In this upper panel, we have two examples of bean seeds. One is of the genus Phaseolus, and the other one is of the genus Ricinus. In this illustration, we can see the endosperm of the Ricinus seed. Note that aside from the endosperm, which is this structure here, the seed also has a cotyledon, which is the structure that you can see in the middle. Compare that with the Phaseolus seed on the left. Phaseolus seeds do not have endosperm, but have cotyledons. It is a structure, this full structure that you see here, and also here. Now, if you look at the lower panel, we have examples of monocot seeds here. Specifically, we have onion and we have corn. From these illustrations, you can already tell that the endosperm thickness in these two monocot seeds differ. Also, notice that onion seeds have a single cotyledon, which is absent in the corn seed. This is the cotyledon in the onion seed, and we have here our corn seed lacking the cotyledon. But instead of the cotyledon, we have a modified cotyledon in the form of scutellum, and that is the structure that you see here. In seeds of grasses like barley, wheat, and rice, the endosperm differentiates into two structurally and functionally distinct tissues. What are these tissues? We have the starchy endosperm and the alurone layer. We have here a picture of a barley endosperm showing both structures. This here in the middle is the starchy endosperm. The cells of the starchy endosperm are dead immature cells, although they contain enzymes that function during germination. Aside from starch, these cells also store proteins. Besides the starchy endosperm, we also have an alurone layer, which is the pink outline surrounding our starchy endosperm. In contrast to the starchy endosperm, the alurone layer is alive in the mature seed. After germination, it functions as a digestive tissue that synthesizes and secretes large quantities of enzymes such as alpha amylase to mobilize food reserves that are located in the starchy endosperm. This slide shows a picture of the same starchy endosperm and alurone layer in barley. You can see that the starchy layer contains starch granules that vary widely in size. So you can see here a bigger starch granule surrounded by smaller starch granules. In the scanning electron micrograph, you can also see a concentric pattern within the granule, which indicates the concentric deposition of starch. Lastly, we have the seed coat. The seed coat, otherwise known as the testa, is the structure that encases both the embryo and the endosperm. The seed coat is derived from the integument, which originally encloses the ovule. From the illustration in the right, you can see the integument structure right here. The main function of the seed coat is to provide protection to the embryo inside the seed. The various chemical composition of the seed coat contributes to its protective functions. The seed coat is composed of one to many layers of dead cells. These cells often contain phenolic compounds such as polyphenols, tannins, and proanthocyanidins. The phenolic compounds are responsible for the brown to red coloring that you see in many seeds, like for example, what you see in sorghum seeds and also in rice seeds. Aside from imparting the distinct brown to red color in seeds, phenolic compounds also have other functions that are directed towards protecting the seeds. For example, some phenolic compounds like tannins and polyphenols give off an astringent flavor to seeds. The astringent flavor is the flavor you get if you try to eat a banana that is not yet ripe. This is the flavor that makes your mouth feel dry and pucker. This taste makes seeds unattractive to pests and actually prevents them from damaging the seeds. Other class of phenolic compounds, the proanthocyanidins, can filter out harmful shortwave light. Proanthocyanidins have double bonds in their chemical structure that allows the compound to absorb damaging ultraviolet light. By capturing UV light, the embryo within the seeds are protected from the mutagenic effects of the short light wavelengths. Aside from phenolic compounds, some seeds also accumulate polygalacturonic acids that are deposited in the outer surface of the seed coat in the form of mucilage. These mucilages hydrate quickly when exposed to water. This makes them important in the uptake of water during seed imbibition. Given that mucilages encase the whole seed, it allows for uniform water absorption over the seed surface. These pictures here are Arabidopsis seeds stained with a dye called ruthenium red. In the left, the seed shows two mucilage layers indicated by the outer cloudy layer, the lighter pink stain that you see here, and a dark staining layer close to the seed. In the right is a seed mutant lacking mucilage. Here are some other examples of seeds showing mucilage. These are dry seeds of tulsi or Oxymum sanctum. Tulsi is also known as holy basil and is used for religious or medicinal purposes. When soaked in water, you can see a thin white layer forming around the seed, and that is the mucilage. In panels A, B, and C, we have the plant, flower, and seed of the cactus Leptocerius scopulophilus. Panel C shows a seed of this cactus stained with crescent blue. The cloudy blue structure surrounding the seed is actually the mucilage. Tomato seeds are also an example of seeds having mucilage. So what we have here is a picture of dry tomato seeds. You can hold a single tomato seed using your 2 fingers when they are dry, but when they are fresh, tomato seeds are very difficult to hold because of the gel-like slippery structure that surrounds each seed, and that is actually the mucilage. Seed coats of many plant species are much more complex because they are fused to the pericarp. The pericarp arises from the ovary wall, which is the structure that you see here. Seeds that are enclosed by a testa and a pericarp are technically fruits. Examples of fruits in which a fused testa and a pericarp surround a single embryo are the caryopses of grasses like your corn. The yellow outline that you see here and that we mistake for a seed coat is actually seed coat fused with the ovary wall. Technically, that makes each corn kernel a fruit. Another good example of this technicality is the strawberry. Each spot that you see here in this picture, the structures that we think are seeds, are actually individual fruits called the achenes. The red fleshy structure is actually a modified receptacle. The receptacle is the thickened part of the stem from which the flowers grow. To summarize, seeds are fertilized, mature ovules that result from double fertilization in plants. The seeds have 3 basic structures: the embryo, seed coat, and the endosperm. The embryo is the earliest form of a plant. It contains the plumule, which is the embryonic shoot, the radicle, which develops into the root system, the hypocotyl, which is the embryonic stem, and the cotyledon or the embryonic leaves. Then we have the endosperm. The endosperm stores the food that will fuel the growth of the embryo during germination. In most dicots, The nutrients from the endosperm are absorbed into the cotyledons during seed development. Lastly, we have the seed coat. The seed coat is a protective casing that envelopes the endosperm and the embryo. Structural variations in the embryo, nutritive tissue, and seed coat exist between monocots and dicots.